All right, so the last panel is Revolutionizing Cannabis Cultivation, Genomics, Microbiome Science, and Pest Resistance Strategies, moderated by Kevin McKernan from Medicinal Genomics.
Oh, thank you, Zach. Appreciate that. So at the end of the day, we felt it was important to have some slides because we haven't had enough slides today. And it's enough panels of us talking about each other. We're going to put some hard data up there. And I want to introduce the panel here first, but we've got, I think everyone knows Soren. Soren's spoken here a few times before. Now at Aurora, I think in your previous life, you were
postdoc at UGuelph.
That's right. That's right. And then Michael, I've just met from UBC, and I'll pass the mic down for everyone to introduce yourself once I'm done giving the brief ones, which is Maria Rojas. I think we already introduced her just a moment ago, so we're wearing her thin on these panels. It's late and sunny out there, so let's remember that. And Punya's been on our podcast in the past. And unfortunately, Zamir wasn't able to make it today, so I promised to put a couple slides up that resembles some of the things that we've been working on together, and we'll get to that later. But let me just let you guys do a better introduction than what I just did, Soren. Go for it.
Sure. Thank you so much. Thanks for having me. It's a pleasure. My name is Soren Seifi. I got my PhD in plant pathology from Ghent University in Belgium. It's pronounced in Dutch as Gent Universiteit. And then I moved to Canada in 2014. I did 2 postdocs at the University of Guelph. My first postdoc was focused on Botrytis arabidopsis interaction, and my second postdoc was focused on breeding for disease resistance in wheat, cereal crops. And that was actually the postdoc that helped me to land this job position at Aurora because they were searching for a pathology scientist with some breeding background. At Aurora, I started working with breeders, agronomists, molecular biologists, working on disease resistant to powdery mildew. And just recently, a couple of months ago, we published our paper on the characterization and mapping of PM2 gene, the second gene that discovered against powdery mildew. And I'm going to talk about it more today. Thank you.
Hi, everybody. My name is Punya Nachappa. I'm an associate professor at Colorado State University. I'm an entomologist by training. My research focuses on molecular plant-insect interaction, insect vector-virus interactions. So if that term is not familiar to you, a vector is an animal, in this case an insect, that transmits a virus from one host to another because viruses need a living host, a cell, to move. I also work on pest management. So I've been working in hemp because I'm in a land-grant university since 7 years, since joining CSU. But of course, there are parallels between cannabis and hemp. And this is my first CanMed conference, and I'm having a great time. And what a great location. So thanks.
Thanks. I'm Mike Deyholos. I'm from the University of British Columbia's Okanagan campus. And my background is really in fiber crops, so flax and hemp for fiber purposes, but the universe has drawn me into this world of cannabinoids, and we're working on phytocannabinoids from non-cannabis species such as rhododendron and even some fungi like cylindrocarpon. But in reference specifically to hemp, we've worked on seed-borne and seed-vectored microbiota and shown how those interact with seed-borne and seed-vectored pathogens, both fungal and bacterial, and shown some suppressive effects of the bacteria you consistently find associated with seeds seem to suppress the fungi that are some of the significant pathogens of hemp and perhaps also of greenhouse cultivated cannabis. And with that, we identified instance of Berkeloomyces, which is a black root rot in aeroponically cultivated marijuana.
Hi, this is me again. I would like to just give you a reminder of I'm a by heart a virologist, and but my first years, for the first 5 years of my career, I work at CIAT, an international center for this, and I work on beans. That's what they specialize on. in many different fungal diseases, doing disease resistance in the fields. And then I went to the University of Wisconsin. I got a master's degree there for plant pathology, and I developed methods there for detection of pathogens using PCR there for Gemini viruses. And then for my PhD, I did at UC Davis very thorough study in potiviruses, but also in Gemini viruses for studying how they move inside the cell, doing microinjections in the living cells, and testing different proteins that the virus produce, and figuring out which protein is the real moving protein and doesn't need a partner or not, and how the interaction happens with the plasmodesmata, all the channels that open from cell to cell in the plant. And then we developed many assays for screening for disease resistant and greenhouses to try to help out companies in California from tomatoes, peppers, cucurbits, and now I'm in cannabis, trying to bring all the knowledge that I acquired and that expertise into the cannabis world and rapidly adjusting to it. Thanks.
So we are going to just step into a little bit of a background on what we're trying to accomplish here, which is on plant pathology and how do we attack this from like a genomics perspective. And Soren was kind enough to give me a few slides to walk us through. And I think I want to just make sure I can advance this for you. Okay, you have in control of that guy right there.
Thank you. Thank you so much. You're pretty familiar with that. But so in my interaction with a lot of cannabis breeders and also other crops, it happened to me that I believe that there would be a little bit of misunderstanding about the exact function and position and meaning of true resistant gene, an R gene as we call it. You probably have heard this idea of gene for gene theory developed by Floor in the 40s. He used to work on Ross disease on flax. And this actually theory talks about that for any avirulent protein coming from the pathogen, there is interacting protein on the plant side, a resistant plant side that will that would trigger immune reaction in the plant, and that specific gene and the protein, the product of that gene was termed as resistant gene, R gene. Now, in any RNA sequencing study on plant pathogen interaction on resistant plants, you will see a long list of different genes being highly expressed. And so the question here is that these are all resistant genes because they're playing a role probably like chitinases, PR proteins, a lot of different proteins, antimicrobial compounds, or phytoalexins, cellular fortification related proteins. No, these are defense-related genes, and these are not the R genes that we're talking about. And on the left side of this slide, you see for all plant pathology, this is a kind of a biblical idea. It's called the zigzag model. Daniel and Jones actually developed it in 2006. That is explaining how this R gene actually functioning in inducing resistance against pathogens. So 3 important levels have been defined here. First, we have the recognition of the pathogen, of the presence of the pathogen that can happen at the cell membrane by receptor-like kinases or in the cytoplasm with these resistant genes or R gene in response to the effectors that are secreted by the pathogen. So these are the proteins that either interact directly with the effector protein of the pathogen or they guard an important protein on the plant side that any change of that protein can be sensed by these people, these guys, at the very upstream of the whole cascade of immune response to a pathogen. And that's why, although we see a long list of different genes being upregulated in a resistant plant, that single gene is the one that switches on the whole cascade, and that is located at the upstream of that cascade. So the PTI here stands for PAM-triggered immunity, so there are pathogen-associated molecular patterns that can be sensed by those molecules at this cell membrane level, or effector-triggered immunity, and that's the response that you get when your resistant gene, the cytoplasm, and you know that these genes are known to have specific domains, nucleotide binding site, leucine reach repeat proteins, and that nucleotide site is the site that interacts with ATP and gets the whole protein activated. So that's the definition of a resistant gene. Now, in the coevolution of plant and pathogens also evolved other molecules that can kind of neutralize this reaction. And they came up with this ping-pong coevolution interaction with new effector proteins that they can actually, that leads to effector-triggered susceptibility. One last thing here, a very new model has been developed by Professor Thardal Christensen. It's called the iceberg model. So the gene-for-gene model by Floor is now rather updated. That was so valuable and actually set the foundation for a lot of other scientists who work in this field. But the newest model, a 2020 paper, and I highly encourage you, if you want to know more about the molecular aspects of plant-pathogen interaction, to read that paper. It's called the iceberg model. For example, in this pan-genome paper, Nature paper, but Todd's group, there is a circus plot where you see the number of resistant genes found in cannabis. And if I'm not mistaken, like chromosome 1 had 250 different resistant genes. So the question here is that why so many resistant genes are located on different chromosomes, like hundreds and thousands of resistant genes, while we just found probably 2 or 3 being published, if I'm not mistaken. That is explained by this iceberg model. Throughout that coevolutionary interaction between plants and pathogens, every time that plants and resistant plant evolved a resistant gene, a pathogen actually evolved an effector to neutralize that, and another resistant gene in response to a new effector, and all of them were neutralized and not working anymore. But you can see them in tandem, typically close to each other on different chromosomes. But what we see in that zigzag model developed by Jones and Dangle is actually the tip of that iceberg. Thank you so much. Sorry, it was a bit late.
I actually want to just touch on this topic because to bring this back to sort of human genetics, plants don't have an adaptive immune system, so you can't vaccinate them and have them remember. So perhaps the theory of what's going on here with all of these dormant or excess genes related to pathogen responses, every time the plant has had to co-evolve around some other pathogen, it has retained a copy of one of these ancient tricks, perhaps. Maybe they're still active, I don't know how ancient they are. But we don't have an MHC complex in the plant. We don't have this type of adaptive immune response where we can run around and remember old antigens we've seen before and go and take them out. So the genomes are finding perhaps other ways to store these genetic goods, if you will, over time.
No, I want to cover that later, so I want to be only a speaker.
So on those notes, thinking of, there are obviously bacteria that the plants are oftentimes in coevolution with, sometimes pathogenic, sometimes not. There's fungi, and of course there's viruses and viroids that we're concerned with in the plant world. We don't see as many viroids in mammalian biology, although very recently Andy Fire's group did publish these things known as obelisks, which are double-stranded circular RNAs that do have some coding sequence in them, unlike the viroids. What other areas of the plant's genome should we be considering when it comes to fighting off viruses and viroids? These may not, would these be these PAMPs? Would these potentially, are there potential genes that we think would fight them off, like PM1 or MLOs or chitinases, or is this just an entirely different molecular machinery that we have to review? Does anyone want to try and take that on?
I'm going to start, but I think she's an expert on this. I think one point that I think needs a lot of research is in the vector side and the vector and the plant interaction. So what genes are really in there associated with that? There's a lot of studies. We've done some of that partially, but some other researchers have done studies where you map directly the proteins of the insect with something as the insect feeds and what part of the gene gets turned on in the plant, you know? So I think the gain that we obtain from that part, such studies can be brought into the genome of the cannabis and then select genes from there that we can focus on to try to turn off. Then maybe we avoid the vector, even though if the vector still feeds, we will avoid transmitting the virus, you know, and then we don't have to kill the virus and we don't have to kill the insect. Everybody's happy, but we don't get the disease. A concept like that. I think that would be one area.
Yeah, definitely. Thank you, Maria. She knows mentioning insect vectors will get me excited. I think in this age of genomics and transcriptomics, I think we need, I don't think I've seen a published study of, transcriptomic responses in response to HLVD and generate a list of targets in different genotypes or strains of cannabis. And in hemp, we're seeing very different interactions in response to HLVD. I'll be talking about that tomorrow. So once we generate some potential targets, we have Mike Peterson who can make all these knockouts for us and go from there. But definitely with the potential insect vectored aspect, that's a common route that insect vector biologists have targeted diseases in the past.
Maybe another idea just occurred to me. You know how, excuse me, pathogens differentiate, like they're biotrophic, you know, like the ones like powdery mildew, you know, mildews that they need the plant. They feed from a living cell, you know. They cannot hurt the cell because they depend on the cell, on the living part. And there are nasty ones like some fusarians, for example. They can be in the middle, hemi, you know, a little bit, but they can become the type of pathogen that cause necrosis or kills the plant cell. The word skip my mind. Nephrotic, yes, thank you. Necrotrophics, yes. So if we create like a system where we group what genes are more associated with mildews, for example, that type of pathogens, versus the ones like the fusarians, you know, the ones that kill the cells versus the ones that don't, and there's a third group that is intermediate, you know. Maybe we can narrow down the pool, it depends on and focus on that to see if we can help in the way that the beautiful example of powdery mildew and cannabis is working, you know, for other ones. I mean, if we were to do this in something like fusarium, for example, that would be a tremendous, incredible help and for cannabis.
All right, interesting. And to your point, there is actually a RNA-seq experiment on HLVD infected cooking, and Kimberly Quinn's team has something to say about that eventually. We'll keep it on. We'll keep it on. I still there. Yeah, I think it was, maybe it's poster scale. I can't remember where its publication status is, but.
It was an REU project, and the student is coming back in August to finish it.
Awesome, excellent. But there's some fascinating things in there. Now, on the point of Fusarium and where some of these pathogens may lie, so part of one of the projects we've been doing with Zamir, he's been sending his samples and we've been doing whole genome sequencing on them. And one of the things that falls out of whole genome sequencing is the microbiome that sits in the background of this. So he's been looking at like stems and roots and flowers and leaves. So we've been collecting this very large database of where are all these pathogens in the plant, and if they're pathogens or not is obviously up for debate, but they're clearly endophytes in many cases when we're looking at them. I do have a few slides that I can share on those as well as to some examples. I don't have the exact screen capture, but Soren had asked me offline, okay, where is trichoderma? People are using this as a biostimulant. Where is it in the plant? Is it hanging out in the roots? Does it ever get to the flower? And you can quickly scan through data like that and see its reads per million distribution and all the different tissue types and it relative to Aspergillus that we test for and what would happen if we turned on testing for that or Pseudomonas, for example, someone else had mentioned. So I'll show you some of that a little bit later, but I didn't want to jump the gun on. I think you had another slide or two that you were going to jump to on maybe trichoderma itself and what we can do there. Oh, there you go. You want me to hit this, or I can't even see it right now. Here.
Yeah, I thought, because we had a very good discussion about trichoderma this morning, about mechanisms that trichoderma can induce kind of tolerance or it's used for disease management against root rot diseases. So I thought that these just made it in a hasty manner. Oh, sorry, the reference is not added. I think it's, I'll mention the reference shortly. This is actually showing how trichoderma can parasitize. So there are 4 different mechanisms that trichoderma can kill these pathogens. And the question in the morning was that because trichoderma is a root colonizer, how do we know that it can kind of colonize endophyte as well? And Kimberly, you mentioned that you expect to see them in flower. And just this morning I asked Kevin to check that on his database, and that's correct. There are a lot of reads of trichoderma showing up in the flower tissue, which is good to know. And in these slides, you can see different mechanisms that trichoderma uses. It's, you can see how it drills actually the cell wall or epithelium and fusarium. It goes inside of the pathogen. That's the mycoparasitism mechanism. It also produces antibiotics, antimicrobial compounds in that petri dish. The strain A actually is a wild type strain. Strain B is knocked out on antibiotic biogenesis, and you can see that strain A is very potent in suppressing the growth of epithelium. And competition is another mechanism that is used because the site of contamination or infection is already being occupied by trichoderma, and there's no place for any other fungi to grow there. And the 4th and last mechanism is that induce systemic resistance through jasmonic acid and ethylene pathway. So that was the slide.
Excellent. And to the point of transmission here, we have someone who's looking at, Michael, you're looking at seed transmission. How many of these different pathogens do you be tracking through clones versus seeds or both?
Yeah, as far as seeds, Aspergillus, Alternaria, Fusarium, just about anything you can think of is on the seed, even after you wash it off. Yeah, penicillin. Thank you.
So even bleaching these things or hydrogen peroxide treating seeds isn't necessarily a sure bet of ridding it of a microbiome transmission. It's some of it, I'm assuming, is inside the seed coat?
Inside, of course, what we call the hemp seed or cannabis seed is really a nut, right? So a lot of it is inside of that shell, and very difficult to remove by sterilization then.
And on terms of tissue culture, obviously tissue culture is designed to remove a lot of this microbiome. I've seen some or heard some conversations with others in this field. are you at a risk once you've cleaned that? You have a bit of a boy in a bubble syndrome there. Like, what invades it afterwards? Do you have to like reinstall a new microbiome after you've done tissue culture to make sure that you don't have Fusarium or Pythium creep up on you?
Excellent question. So we start clean because we meristem them, right? So our clones, so they're clean. But then we have to grow those babies. And believe it or not, even the rock wool where we grow the plants, there is penicillium, aspergillus, alternaria, and even fusarium. Amazing. So you've done all this incredible work having to grow these meristems, and you put them in there to get contaminated, you know? So when the plant is growing, they're gonna acquire it through the roots and they're gonna go endophytically. And I'm gonna cover that tomorrow. I'm gonna show you how the insides. So we end up autoclaving the rock wool because I know that Fusarium, which is the one that is the most concerning for us, ain't gonna survive that, you know? So we autoclave the rock wool, and it's extra labor, extra cost, but we are committed to have clean, healthy plants to the customer. So, and then the other thing that we do right there, you mentioned trichoderma, so you can use biologicals, and that's gonna give you a free pass during the first stages of growth of the plant. So anything that is allowed in California is very strict. So whatever, and I'm not gonna advertise any company, but I would recommend a mixture of bacillus and trichodermas, you know? And whatever the name of the product is, test it. But if you protect those little roots growing in there with biologicals like that, they'll give you a fair chance that your plant is gonna be really, really stable and good. There's other things that you can use, like enzymes, like hydrozymes or stuff like that. That's gonna directly chew in the roots, supposedly. But if there's mycelia growing in there, it's gonna chew it up too. It's food. So, and then you can also control the growing conditions so that you don't favor for the growth and the sporulation of the pathogen. Your idea in there is avoid at all costs sporulation of the pathogen. And so if you do that, then, we learned the hard way, but we learn now. So we have in place a very clean start. We use biologicals, autoclave the rock wool, and smile at the plants so they will smile back to you.
So this was a question that came out of the testing community for us, which was, okay, how is this aspergillus stuff getting in? Is it all over the place? Is it in the air? Is it in my hair? Is it everywhere? It's a bit of a Dr. Seuss theme going there. And so we went through all this data and started asking that question. And it's really clear. Now, you probably can't see these slide very well from their distance out there because I can't see it from here. But this is some waterfall plots looking at the concentration on a reads per million basis of Aspergillus nigrotarius flavus fumigatus in the roots versus the stems versus the leaves versus the flowers. And what you can see is its log scales higher in the roots and the stems than it is in the leaves and the flowers. And so that tells us that this is predominantly coming in through the root system. So having clean substrate is actually really important here. If you're not testing or looking at that or sterilizing it in any way, you may be actually introducing this through the fertigation systems or through the actual the substrate that you use. But we can now draw these plots on all the microbes in the database to look, okay, maybe this one comes through the roots. What about things that come through the air? Is there anything that's more concentrated, like botrytis is probably more concentrated in the flowers than it is anywhere else, just based on my visualization of flowers that are infected by it. You might see the opposite correlation in the data there, and that might give you more information on how to handle these. Now, I'm curious on sterilization. I'm sure we'll hear about this tomorrow in other talks, but Rockwool, you can autoclave. What about HLVD? Can you add bleach or anything else? RNase-3 or something?
Well, HLBD, there's minimum, I can tell you, because we do not intake anything with HLBD. We do a thorough test, and we avoid at all costs to bring it into the facility. So far, no cases for us. So I mean, I can speculate if you like to, or she works in HLBD, so she can give you some ideas.
Gosh, HLVD, I mean, you just say the name and it's transmitted to a healthy plant. We thought TMV was bad. And I'm sure you all are aware of how highly seed transmitted and pollen transmitted it is in water. And trying to show that it's vector transmitted, we had to be doubly careful. So again, we autoclave our rock wool, the soil, the water, all different. And we've got different rooms for infected plants, healthy plants. The insects are in different rooms. I think testing, keep testing, try to catch it early. And if we have any inclination that, since we know it's vector transmitted now, and I'll share the data tomorrow, we just pitch the plants, autoclave the plants, end of story, start new. We've got a clean mother plants that we started from tissue culture, healthy clones. If you have any inclination, we just get rid of it. We're testing phytohormones. We heard a lot about these, not microbial beneficials, but other elicitors of the induced systemic resistance or the SAR and the SAR pathways. So those, as long as you fine-tune the timing and the dosage, can be effective. But I think HLBD continues to be the bane for the cannabis industry. In hemp, we have not picked it up since our first report in 2019. Multiple metagenome surveys, field surveys, and we've got numerous field trials. We haven't seen it in field-grown hemp, even though it is highly seed transmitted. And I think the group from Italy, their metagenome did not report HOVD there as well. So seems to be mainly indoor cannabis cultivation. All the clone, yes. I'm sorry, cloners.
Well, there was some history in the cannabis field, and we've been, guilty of this as well, of people trying to graft cannabis to hops to get very bizarre phenotypes out of it. And it does produce some bizarre seedlings. We don't really understand why. We did do some sequencing. We have a paper in review right now about this, showing that there's no type of genome fusion between hops and cannabis, but there does seem to be a very high mutation rate when you do this grafting process, and the seeds come out with just a higher C to T transversion rate. And but I think people working on that may have been the source of this type of transmission between the two species, where we've seen Hoplite and Vari jump over, because the sequence that we see in HLVD hasn't really diverged much from the original two that Warren has published. And he may have some RNA-seq data too. I think I saw something from him very recently on that front. But anyway, on that note, what are the most promising things that we're seeing given some of the new papers that have come out from Todd's group and others on trying to utilize genomics to drive yield and pathogen resistance? How does it get deployed today, and how do you think it's going to get deployed in the future? Do anyone care to take on that question?
So I was reading a paper. We had this conversation call today about learning from other crops and trying not to be very isolated and focusing only on cannabis. And the MLO gene actually was known in other crops. And we knew, before discovering MLO, we knew 100% that it exists in cannabis too, which we didn't know where it is, and what finally found. So in the world of viroids, potato tuber spindle viroid is a very important viroid that attacks tomato and potato crops, and a couple of other crops as well. And it's been widely studied. And it's very, very interesting that these viruses are very smart. It's just a naked, single-stranded RNA molecule but just listen to this story. It's very interesting. It can hijack, of course, we know that PSTVD, just like HLVD, they belong to a POSB viroidae family. They go to the nucleus, they hijack the RNA machinery of the plant, and they replicate themselves. But not only that, they hijack the anti-viral resistance response of the plant as well. And if you're familiar, these dicer-like enzymes, they try to chop the viral genome. So they detect some highly structured region of the genome of the viruses, and they chop that. I mean, they chop that, and the risk, the complex risk, detects that those 21 to 24, 25 nucleotides, short reads, and then they start actually degrading the genome of the virus in an HLVD or PSVD. But by doing that, those small areas that are highly structured, for example, in potato spindle viroid, are also highly homologous with some defense genes of the plant. So the pathogenicity region is called on the viral genome. The pathogenicity region is highly structured, and that is the region that dicer-like enzymes attack, and that is the region that is homologous with some important defense-related mRNA of the host. So by reacting to the viroid and trying to neutralize the viroid, the host is actually neutralizing its own defense genes. And the genes are like, one of them was Deyholos synthase gene that can block the plasmidesmata in the phloem because the virus actually travel through the phloem. So that is downregulated already. The plant is trying to defend itself, but it's actually neutralizing its own arms. Another gene was related to PR1 proteins. Another gene in cannabis, Dr. Punja showed that there are three important sequences in HLVD genome that are highly homologous with some important genes in cannabis. One of them is COX7. Another one is calosyntase, expansin is another one. So this stunted growth that you see, it's actually it's the product of the defense response of the plant trying to degrade the HLVD genome. Yeah.
That picture for you right there. So this is a picture of HLVD, and it's color-coded for those regions that that Soren was just mentioning. So the three genes that HLVD has the most homology, sequence homology to. These are short homologies, or 19 bases, but that's about as good as we can get. 19 to 20, I think, are expansin, calosyntase, and I think COG7. So you can see some of the structures. Now, the reason we're doing sequencing of these viroids is if you ever get a mutation in one of those spots, the structure can completely change. And then maybe it targets a different region of the cannabis genome. And if there are regions in other genomes, cannabis genomes themselves, where there are mutations in these genes such that this thing isn't homologous to it anymore, it may lead to a different form of, I wouldn't call it resistance, maybe tolerance or something, right? Where it may not be as effective in that RNA interference pathway if there's differences in the viroid versus the genome. So we've been trying to track these things on Cannapedia and Viropedia to see if anything emerges. We haven't seen a whole lot of variation, though. That's the thing. I know initially a lot of variation was reported on HLVD in some of these thermal mutants, or putting them in different plants. But when we've been surveying what we're finding in the cannabis space, let me just say they're hard to sequence. Sanger sequencing, I think, makes a lot of mistakes on these things because they're hairpins, and so there could be some noise in the database due to that. But also, when we go through this with a lot of deep shotgun sequencing with Illumina, we don't see a tremendous amount of variation outside of like CAN 1 and CAN 2. So that's an area we're hoping genomics will maybe shed some more light on. Is maybe we'll find some tolerance out there, because clearly in the hop field hasn't gotten rid of this. They have found plants that don't have a yield loss due to it. And I think that's hopefully where we'll end up in the cannabis space, is we'll find plants that effectively can tolerate it to some degree. I don't know if that, if you want to comment on that.
I was going to comment on another aspect of HLVD and hijacking. This is for the potato spindle tuber viroid. To get transmitted, because this is such a simple, pathogen, to get transmitted, potato spindle tuber viroid hijacks the coat protein of another virus that co-infects the plant. And then it's able to get into the insect, the aphid stylet. For a virus or, to be transmitted, it has to attach to the mouth part, which we call the stylet. And so we're wondering, you were mentioning this, Soren, and we know the laundry list of viruses that are in cannabis and hemp, and we don't know what it is they are doing. Cannabis sativa mitovirus, cannabis cryptic virus, and we always find them. Every single metagenome survey we've done, we find them. So maybe there is this interaction going on for them to be transmitted, because HLVD was not transmitted by hops aphid. So a lot to learn in the cannabis virology space.
That I should forward you the list that were. So when we do this DNA, did I forward you the? Do any of those make sense? Because I don't know much about DNA viruses.
Kevin, yeah. So metagenomics is, I don't know if I should say open a can of worms or Pandora's box, but has a list of DNA viruses. Maria, you'll be interested. Most plant viruses are RNA viruses, but Gemini viridae, one of the most notorious ones, beat curly top, yes. But I was surprised to not find any of these Gemini viridae viruses in your DNA virus list. So most of them looked pretty novel, but again, we don't understand right, exactly. Unless we do some Koch's postulates testing, we won't know, yes. Did you want to?
Okay, well, we've got about 15 minutes left here, so I want to make sure we can get the audience involved here with questions so that we can help perhaps guide this a little bit. And I see lots of hands up, so there we go. Zach's going to run you with the microphone. So the methods that we're using are whole genome shotguns. We only capture the DNA viruses. By complete accident, we just found those in blast hits and don't know what they mean.
So my question was about if you guys are creating resistance to pathogens. I know that a lot of beneficial microorganisms like trichoderma, mycorrhizal fungi create similar molecular patterns to bypass the plant's defense system when they're making these relationships. Have you looked into the ability for the plant to make those types of myco-associations when you're breeding in resistance to pathogens?
So we did some work in flax with co-inoculation and separate inoculation of Fusarium and of arbuscular mycorrhizal fungi. And the plant responses were very, very different, even before colonization or attachment with fungi. So I think that the plant is able to, plants have already evolved to be able to distinguish between those beneficial fungi and the pathogens and shut down the appropriate pathways. So I think there are clear targets, at least in the, say, RNA-seq data we did in those responses that are very specific to the pathogen and yet allow the beneficial fungus to still colonize. So I think the targets are there, and it's possible to find genes that you can suppress to suppress the pathogen while still allowing the beneficials to do their job.
Yeah, great panel. I want to go back to kind of midway through the conversation. There was a really fascinating discussion around the natural microbiome and how in the absence of that, pathogens may be more inclined to proliferate. And I think what you said, or what I'm hearing, is that there should be a baseline level that is inherent and exists to protect the plant.
Yes, and an important point in that, Chris, was the mention of Koch's postulates that you brought up. And so I think what you're hinting at is the total yeast and mold testing we have in the industry, which may be harming the ability to use some of these beneficial microbes. We cannot get through Koch's postulates. because we don't know what it is we're measuring with those tests. So this is why there's a dearth of clinical literature suggesting 100,000 CFUs might be safer than 10,000 or vice versa. It's not something we can prove. So we're stuck not having any literature to guide regulators on, yeah, maybe 10,000 is the right number, maybe 50, maybe a million. We don't really know what is the harmful amount of TYM on a cannabis plant. This is one reason we've been trying to push for species-specific testing, because we can do those things. We can isolate Aspergillus, infect something, and demonstrate that it's in fact infective. We've actually done that now with HLVD. We took HLVD and chemically synthesized it with GenScript. So we have a perfectly pure RNA. There's nothing else in there that could be contaminating it. We can take that and inject it into clean plants and then watch it proliferate throughout the plant for 2 weeks. So we have Koch's postulates like nailed for that one, at least in the infectivity of plants. But we really need to be able to do that for all the other plant pathogens that the regulators are asking us to target. That, most of the ones that lifted over from food, that's already been done in the food industry. So E. coli, Salmonella, if it's pathogenic on beef, we don't need to reprove pathogenicity on leafy greens. It's assumed to still be pathogenic on the two matrices and thus get superimposed on the cannabis. We have much harder time establishing any of that pathogenicity on TYM. So I do get nervous when those tight limits are in place. I think it was Illinois that may have had them at really, really low levels that I don't think anyone can accomplish unless they irradiate. What is that doing to the breeding market in Illinois? They can only breed plants that have to have genetically a very low level of microbiome. That's going to probably create a bottleneck or force everyone into decontamination. Zamir, who's not here, did have a nice paper showing that there is a genetic basis behind the plants that have high and low TYM counts. And I think on that scale is like a factor of 20 or 50-fold difference between like his powdery mildews versus I can't remember the other plant that was really low, but he had one that was very low and powdery mildew was very high, or powdery donuts, sorry. And so there is a genetic basis to this, but I don't know that there's a plant out there that can always sustain less than the 300 CFUs or something they had in that Illinois regulation. So this idea of breeding a plant that's completely pathogen-free, I don't think is ever going to happen. We need to have, I think, more sensible regulations there that are targeting things that can get through Koch's postulates.
I'd like to add one important point. That is, let's say you send something, a third-party lab to get tested, and you get the result back and it said, positive for Fusarium, and you freaked out, pretty much. But I'm here to tell you, not all the Fusariums are pathogenic. And not all the Fusarium oxysporins are pathogenic. So that's when a pathogenicity test or the Koch's postulate is huge for you to get a better understanding of what you're dealing with. It could be just a Fusarium that so happened was in the soil and the media that you grew the plant, and it's just hitching a ride, just living in there nicely, and it's not even planning to attack the plant and make, So, but unfortunately, when you extract the DNA and do a PCR or qPCR, whatever the company is using to do your testing, doesn't distinguish between that. It's only going to read it's Fusarium, and it's going to give you a positive. And honestly, that needs to be taken as a false positive. So education is critical here. Education for the cultivators, education for us to understand, okay, so I don't have to freak out if I get a positive for a Fusarium. So which Fusarium am I dealing with? Is it proliferatum? Is it oxysporin? Is it verticilloides? What do I have? Or what families do I have? Are they coming together and then they're going to attack? Or is the plant under stress? I'm putting the plant under stress because environmental, or I have HLBD or something, and Fusarium comes over and take over. Because one of those that wasn't even planning on being pathogenic, wake up and it has to defend itself because I have to make a living. So it's a very important point.
I had one slide up here to address that point, and Kimberly had asked me about this because I don't think it was very clear on our slide out there. But so what we've been trying to do to address pathogenicity of these is we've been taking the whole genome shotgun data and assembling the stuff that doesn't align to cannabis and then running it through a program called PathoFact2, which looks for antibiotic resistance genes, virulence factors, and toxin genes to just weight, like what is the frequency of these types of hits in the whole genome sequencing data to maybe drive to that point. And I just wanted to give a bit of a shout out to Zamir here because his samples came in the cleanest actually up in now we're comparing him to the hemp world, so this is not necessarily a huge hurdle, but it's these were the other samples are from hemp that we got online, and as many people know, there's really no testing requirements, and many of the samples that presumably fail the regulated markets perhaps wander into that market. And we also look through some of the Massachusetts dispensary data to show they're somewhere in the middle between those two. Massachusetts is known to not test for Aspergillus, which does have toxin genes, but it does test for things that are not necessarily pathogenic. They're just total yeast and mold and testing in the state. So anyway, that's something that I think in the future, as we get better metagenomic tools like this, we can perhaps get a better gauge of, okay, is the Fusarium you're dealing with, does it have toxin virulence genes and pathogenicity genes in it, or are we dealing with a commensal that's not to be worried about? Any other takers out there? Yeah.
Sorry, very briefly, the images of trichoderma that you saw in my slides are from Hubbard et al. Phytopathology, 1983. So, needed to say that.
Do you want me to forward that slide, or was there a... You had a couple other ones in here you didn't hit on. We may have time. Let's give Kim a shot here.
Yeah, I've got a question because I'm thinking about this project we're doing, and you just said that the HLVD hijacks the plant, produces the plant RNA. If I'm looking at transcriptome data, am I looking at what the virulate has made, or am I looking at the plant?
Good question. So that was on potato tuber spindle virus, not natural VD. I just read a paper on that. They showed that there are two genes controlling dicer-like enzymes in tomato against PSTVD, DCL2 and DCL4, if I'm not mistaken, and yeah, dicer-like enzymes. And these are the ones that, when they're overexpressed and upregulated, induce resistance against a couple of different viral diseases. But when they're upregulated, they induce susceptibility to the viral pathogen. So meaning that when the antiviral system is highly activated, it's actually proving that can kind of neutralize the plant response because the plant is actually silencing its own mRNA related to the defense response to viral diseases. So that was coming from that paper, not related to HLVD.
Okay, thank you.
You're welcome.
So I was actually expecting to see in that data that callosynthase would be up or down regulated to some statistical significant degree because that dicer would come in and cleave the region of all the transcripts that are related to that. But I don't know that we saw that in the analysis your student did. So
I don't remember that, but we only looked at, in that short period of time, we only looked at what was in the roots and in the meristem. We haven't looked at the rest of the plant.
Okay, yep, good point. It could be in the flowers and yep.
Was it a resistant or susceptible variety?
So that was a critical flaw that we didn't recognize until the end, that this plant seems to keep it in the roots. But we only learned that through lots of PCR and RNA sequencing at the start. But it did, in fact, and really at late stage, late growth, we could see it getting into the green canopy, but the plant turned a deep purple everywhere else, and we couldn't detect the viroid in any purple tissue. And I don't think it was an anthocyanin inhibition of the PCR, because we took the purple tissue and tried to spike it into other positives, and we can get the PCR action to work in the presence of the anthocyanin. So I think there's something on the anthocyanin pathway that's kicking on as a response to the virus that keeps the viroid level lower than we can detect in PCR. So yeah, there's a wealth of stuff in that data, but now we know we really need a control plant, one that we know gets horribly sick from this thing, and one that perhaps is somewhat tolerant to it. Okay, I think we are Well, we got time for maybe one more question if anyone's curious enough to ask. If not, we can turn it over for the next meeting. Kim, please.
The questions. When you're talking about looking at the taking the plant tissue culture, getting the clean plant, then going in and autoclaving the rock wool and then putting in the biostimulants, can you also just use the biostimulant in the presence of an unautoclaved rock wool? Is that sufficient for protection?
Depends on the pressure of the pressure level of the contaminants that you have. If you only need one microcamellia, let's talk about Soren, for example, to be in the perfect environment to grow mycelia and sporulate. Once you hit that, you're done because it's thousands of spores everywhere, right? So we don't need, you don't really need a high CFU for those to start with. So yes, depends on how many you have. You can use the bioinoculants at the beginning, and your pressure is low, you probably will be okay, and you're probably going to protect the roots, and you probably in that Fusarium is not going to make its way through the roots up in the plant. But depends on the pressure level. You get hit more than once, or you have wounds, and that not only do depend on the uptake through the roots, but through a wound, then you could be in trouble. You know?
Sorry, Jack, I can just speak up. Just a quick one. You mentioned. You just briefly mentioned about the purple tissues having a lower viral load or lower presence of the virus. Can you expand on that?
So they had in this particular plant, it's a Jamaican lime plant that was infected with a viroid and a hop latent viroid, and we dosed it with one that was synthetically pure. It came from synthesized RNA constructs. So there's no like fusarium or pythium like contaminating this type of experiment. And then when that happened, we were then tracking it tissue to tissue with qPCR and then doing RNA sequencing on all those tissues every single week for a series of weeks to track the infection throughout the life cycle of the plant. And this plant seemed to keep it in the roots for its entire life cycle until extraordinarily late in flower. We could start to pick it up in the canopy only in green tissue. But the majority of the plant went really dark purple compared to its control. We had a control that we didn't infect. And that one didn't go deep purple. The one that was infected did. And any tissue we sampled that was dark purple, we could not find the viroid in it. And initially, we just thought that was anthocyanin inhibiting the PCR signal, or it's a pigment, maybe it's screwing with the fluorescence. But we've then gone back and taken that pigment and put it into other PCR reactions to show that it's not inhibiting a positive control PCR reaction. So it's more likely an immune response from the plant during the viral infection that might be playing some role in inhibiting its progression through the canopy. That's a hypothesis, not proven, but we've seen it listed in other viroid studies. If you look through the potato spindle viroid, tuber spindle viroid, it has some connections to the anthocyanin pathway as well.
Thank you. Yeah, we'll take any glimmer of hope, I guess.
Just to clarify one thing there, that's fascinating. But remember, some strains also have purple stems, so don't think that something's going on with HLBD either, right? So yeah.
Oh yeah, but I didn't mean to say that's causative HLBD at all. Yeah, it just seems as if some plants that are tolerant to it, they may be tolerant through stimulating the anthocyanin pathway. That's my guess. I think I saw. Well, what do we got? We're over the time. Is it 5?
Any more questions?
All right, Zach. So take over, Zach.
We got one more, Kevin.
So just a different perspective on sterilization of rock wool. Has anyone considered use of a short-lived facultative anaerobic such as lactobacillus as a means for biological sterilization, and then implementing a microbiome that's been facilitated from the plant's endemic natural system and compared that to, sorry, I'm getting a little ahead of myself here. In the Korean natural farming system, they use lactobacillus as a pre-treatment before they inoculate with indigenous microorganisms. And I'm just wondering if the indigenous microorganisms from an endemic plant and the endemic microorganisms would prevent the infection from an outside viral presence because it already has the microbiota to facilitate all of the means for metabolism of secondary metabolites from those connections or interactions in the rhizosphere.
I didn't quite hear everything you said, but I think I understood. Are you asking me for the role of endophytes in the plant? Whatever microorganism is inside the plant already, can they fight them?
On the sterilization of rock wool as an alternative to autoclaving, like the meat industry uses.
Biologicals.
Using a facultative anaerobic such as lactobacillus. And then doing cross-analysis from the microbiomes that are grown in facilities compared to native regions and the endogenous associated microbiota that's associated in the rhizosphere. Because we're taking these land races out of their associated soil profile as well. So there's a lot of biogeological factors that are also associated with the facilitation of interactions and also the plant sending out chemical messenger signals to basically essentially harvest communities of microorganisms that will help facilitate in procuring the resources necessary for creating those secondary metabolites that will inhibit the pathogen infection to begin with, the preventative means.
So I'll let Maria answer, but I think your question is there a benefit to using a more complex or more natural community as an inoculant rather than commercial bacillus bioinoculant for sterilization or for disease suppression on the rock?
Yeah, also the cross comparison between endogenous cannabis populations versus cultivated, industrial cultivated pathogens and relationships.
I think when you are in an environment like tissue culture, you don't have the luxury of growing your plant in soil where you can find all the natural bacillus, all the species, all some other things that the plant acquires in like the human bio, right? You have good bacteria and not so good bacteria. So in a regular environment like that, that's more balanced, I guess. In a system like mine, like the one that we have, Rocco's sterile, doesn't have anything. And so we have to feed the plant with something, and we use it as a mean of protection, anticipating, no? We're not intentionally feeding it just to create a different microbiome. You know, I am almost sure that the little plant has their own microbiome. It's not stripped from it, you know? Not 100%.
But if the populations were incomplete or out of balance, then could it lose those gene expressions? I guess what I'm trying to say. Yeah, sorry, I just lost my train of thought.
We can talk some more. Try to understand what you're asking.
I'm very autistic.
Well, so I think the question was whether inoculating a substrate with the more complex bacteria that would help cleanse it out and then reintroducing another one, which I think is a totally valid idea, there are ways to do this, that it may not just be autoclave and then add in, one species. Perhaps there's a more sophisticated way to go about doing this. So,
I think the two references that I am speculating from is the research that has been done in fecal transplants in humans, and also in studying E. coli in an isolated environment versus in it's an endemic environment.
okay, yeah, that makes. I think I'm familiar with Sabine's work as well on that front. So there's some work that has to be that. Well, we should talk afterwards, because I think I know where you're going, but I don't exactly know what to do. And you may have some ideas to fill in the holes there, which is not to rely on just pure sterility of autoclave blasting this, but maybe there's a way to sterilize microbiologically,
Right.
and then swap it.
Right, because we still aren't familiar with all of the factors that are causing epigenetic changes, and we're still also figuring out how to do the bioinformatics and analysis of the expression of genes to phenotypes, correct?
Yes, yeah, it's certainly worth it.